Hello. In this session, I am going to talk about gene transfer in plants. Plants are a diverse population. So the methods we are using for one plant may not be efficient for the other plant. So we have a variety of methods that can be used in plants, and we should take or we should choose the method depending on the type of plant we are using for the genetic modification. Further, genetic modification and creation of transgenic plants has special importance because we are using a wide variety of genetically modified plants in agriculture, just like the herbicide resistance, pesticide resistant, salt resistance, disease resistance, etc. So here is a list of methods that can be used for introducing genes into a plant. This includes chemical methods, electroporation, particle gun, liposuction, micro and macro injection, pollen transfer, delivery via growing pollen tubes, laser mediated gene transfer, and fiber mediated gene transfer. For the most of the methods, principles and instrumentation remains the same, just with the bacteria and animal cells. In the following session, I am going to talk about important methods and the exceptions, why how it is differ from gene transfer in plants, in animals and bacteria. So the chemical methods, we can use different chemicals to improve gene transfer in plants, which includes polyethylene glycol or PEG, polyvinyl alcohol, and calcium phosphate. For PEG and poly calcium phosphate, the principle is same with the animal cells. It will precipitate the DNA on the surface of the plant cell, and then the plants will take it up using endocytosis. And if you are using synchronized protoplast for this chemical methods, the results will be better. And most of the experiments will be using protoplast for this experiment. After gene transfer, we need to grow the cell and make the calais, and from the calais, we need to develop the plant. And I'm sure that in your plant biotechnology sessions, you are very much familiar with that. Next is electroporation. The instrumentation and all everything else is similar with the bacteria and animal cells. The electroporation, in case of plants, we can use two different methods. One is low voltage long pulse method, and secondly high voltage short pulse method. So some plants will respond with the one method, and some other plants will be responding with the next method. So it's depending on the type of plants that we are using, and this electroporation is very much successful in petunia, tobacco, maize, and rice. Then in order to improve the efficiency of electroporation, we can either use a heat shock before electroporation, and we can add the PEG into the medium to improve the efficiency of electroporation. So if you are inserting using the intact cells instead of like protoplast, mild enzymatic treatment before the electroporation is also useful to improve the efficiency. Then the particle gun or the gene gun. For plants, we are using 1 to 2 micrometer diameter gold and tungsten particles as the micro propeller. Then we can either do the particle gun in a tissue culture, or we can do in a whole plant. So in the whole plant, the picture shows that how it can be done. For the protoplast, we will isolate the protoplast from the plant, and this protoplast will be subjected to the gene gun or the particle gun. Then it will be grow into the callus, and we will actually go for the shoot induction and root induction, and after acclimatization we will get the plant. So that's how it's work. Then we have one method called fiber mediated delivery. This is an extension of micro injection, but in this case we are not injecting each cell separately. Instead, we are using capillary tubes. The capillaries made up of silicon. These are very small capillaries, and we will mix these capillaries with the DNA solution. So the DNA will be inside this capillary, very small capillary but very sharp. Then we will mix this DNA silicon capillaries with the cell. So while we are mixing, it will make random holes in the cell membrane, and it will actually, due to the capillary action, it can inject the DNA into the nucleus of the plant cell. And this method is a crude method when compared to the micro injection, but it works for maize and tobacco. It is very easy to perform. Next is transformation via the pollen tube. This one can be done for some plants, Uh, During the time of pollination, pollen grains will be deposited in the female flower part, and from there it will grow a pollen tube, and this pollen group reaches the ovary, right? So you know that. So after pollination, what we do, the after pollination and the creation of this pollen tube, we will cut the flower in this area, and we will add DNA here. So along with the male gamete, the DNA will transfer to here, and it will end up in the ovary, and the zygote which is formed from this particular plant will also have our DNA. This method is also applicable for very few plants. Next is my macro injection. In the macro injection, we are using the bigger syringes that we are using in the hospitals and all. So but what we do, we are actually injecting the DNA into the developing inflorescence. So some of the cells are able to take up the DNA in the inflorescence form. So we are just giving an injection, just like we are injecting humans. So some plants like Ray can do it, can take up the DNA like that, but the frequency of transformation is relatively low, but the technical expertise needed for this method is also low. Next is DNA uptake by mature zygotic embryos. Some of plants have zygotic embryo which has a tendency to take up the DNA from outside, especially in the dry condition, which includes wheat, barley, and pea. So the seeds will be dried, and we will incubate in a solution containing DNA. So this dry seeds will take up the DNA from the medium. So that's a very easy method. Uh, so these are the methods we can use specifically in plants, and all other methods is also applicable for the plants because of the diverse nature of plant cells or plant tissue. We need different methods. Thank you.